what we're going to do this morning is to talk about some sort of practical aspects of haematology, so a little bit about how the analyzers work and what all these numbers mean. And then we're going to talk about how the blood film can help us to make a diagnosis in anemic patients. And we're going to use a few cases along the way to help illustrate these techniques. So just to stress, really, I realise that in busy practice, often a blood smear isn't examined, but really in an ideal world, one should be examining the blood film, and particularly if there is an anemia or altered white cells, it becomes essential then. So just some basic guidelines in how to run the haematology properly. It sounds obvious, but it's really important that the sample's mixed carefully before you run it. This is just a rotating mixer which spins round and round slowly to make sure the samples are mixed. Otherwise, one could just invert the tube several times before running the sample. But if you don't mix it, you could have very considerable errors, because obviously all the red cells will settle down to the bottom of the sample and the top half of the sample will be mostly plasma. And depending on which bit you aspirate from that tube, you could get very seriously erroneous results. So just very important to mix it first. And then the next thing that's very important before we run it is to check there's no clot in there. So you're going to ideally use a wooden little stick, which is known as an orange stick, and you just put 2 sticks in the tube and you sort of wipe them round the edge of the tube and then pull them out, and if there's a clot in there, it'll stick to the wooden stick and you can see it very easily coming out. It's really not sufficient just to look at the tube with the naked eye. Sometimes a small clot wouldn't be visible and you wouldn't detect it unless you did this. Also, you can have a look to see if there's gross agglutination in the sample, and if it's been sitting down and settled out a bit, you can have a look to see if it's very hemolyzed. If there is a clot in the sample, even a tiny clot, it will have some dramatic effects on the counts. And as I'm sure you know, the platelet count will be the main one to suffer, so that will be markedly falsely reduced. But also the white cell count is falsely reduced if there's a clot present, because the clot basically is an amalgamation of lots of platelets and white blood cells. Sometimes, in fact, there is a very slight increase in hemoglobin and hematocrit if there's a large clot, but that's not as significant as the white cell count and platelet changes. So then we're going to, having checked our sample quality, we're then going to run it through the analyzer. So just to briefly mention the types of analyzers we have impedance analyzers and flow cytometers, and also there are QBC analyzers still around, although these are much less used now. So on the top right, we can see an impedance analyzer and then the ProCyte IDEX flow cytometer analyzer. So then just to mention a little bit about impedance analyzers first of all. So there are many different types on the market, so I've just listed a few here that are available. I don't have any comments on which particular one is better than the others, but they all work in a similar way. And so we have 2 chambers which contain electrically conducting fluid, the internal and the external chamber. So here's the internal chamber and the external chamber. And there's a small hole here where the fluid can pass through, and therefore the electric circuit is complete because of this hole. So the fluid, electrically conducting fluid, is passing through that small aperture. And then the cells are directed at the aperture, and as they pass through the aperture, they interfere with the electric current. So there's a small blip each time a cell goes through the aperture, and that's how the presence of a cell is detected and counted. So the number of cells present is determined by the frequency of the blips or pulses, and the height of the pulse is what is used to calculate the size of the cell. And the analyzer then simply works out if it's a platelet or a red cell or a white cell from the size. So you can see on the left we have platelets, which are quite small, and then a nice big discrete peak, which is the red cells, which are larger. So providing that the platelets and the red cells don't overlap in terms of size, then these can be counted properly. So just in a nutshell then, the cell size is measured from the height of the pulse, and so that's used to calculate the MCV. The platelet count and the red cell count and the white cell count are measured from the frequency of the pulses. And what the analyzer will do is take an aliquot of cells and lyse the red cells, and then again pass it through the aperture and count the white blood cells. Another aliquot of the blood is taken to a different part of the analyzer, and the red cells are lysed. So there's now free hemoglobin in the solution, which is then measured with a biochemical method using spectrophotometry. And then these other parameters are calculated. The hematocrit, mean cell hemoglobin concentration, and mean cell hemoglobin are calculated from the other parameters which have been measured. So here are the calculations. Don't have to remember these formally as such, but just remember what goes into them. So the hematocrit is a product of the cell volume and the number of cells. The mean cell hemoglobin concentration is the hemoglobin divided by the hematocrit. And as we've just said, the hematocrit depends on size as well as number of cells. Whereas the mean cell hemoglobin is calculated from the hemoglobin divided by the red cell number, but not affected by the red cell size. So what are the limitations of these analyzers then? The first big limitation is this matter of accurately counting platelets, because it's a matter of where the cutoff is made in terms of size. So if you have large platelets, they can be falsely counted as small red cells, and if you have small red cells, they could potentially be miscounted as platelets. And now in cats do have quite small red cells, and they often have quite large platelets, and so you can see there the error is going to be more frequent. And so I'm afraid these analyzers aren't necessarily very accurate at counting platelets in cats in particular. Here's just an example of a clinical case where we had some curious results which we had to think about. Was there a laboratory error causing the changes? So it was just a Springer Spaniel that came in with very clinical signs of exercise intolerance, lethargy, and mucosal pallor, and an enlarged spleen were found on examination. So if you look at the results, then we've got some very curious results here. We can see that the hemoglobin is about half of the bottom of the reference range. So instead of 12, it's down to 6. Whereas the red cell count is within the normal range. That's very unusual. The hematocrit is perhaps down to about 2/3 of what it should be. So none of these things tie up. These should all be reduced by about the same degree. Red cells, hemoglobin, and hematocrit should all go down, in tandem with one another. And so they haven't done that. They're all decreased or altered in a very different way. You can also see that the mean cell volume, the average size of the red cells, is really tiny. It's almost half of the bottom of the reference range. And likewise, the mean cell hemoglobin is very low. But the mean cell hemoglobin concentration is within the normal range. Remember, that's determined by the red cell size as well as number. And because the red cell size is low, perhaps that's why that's normal. So what could be going on that could cause these changes? It can't be real. It doesn't make any sense at all. Let's have a look at some more parameters that we obtained from this patient. The platelet count from this patient was over 3,000. And here's the blood film to show you all these platelets. So here's the red cells, and there's huge numbers of platelets, some of which are very large indeed. So basically, these large platelets and the above average ones are being counted as small red cells. And so that's why the red cell count is higher than the hemoglobin or hematocrit, because actually it's not counting red cells, it's counting platelets and red cells and calling them all red cells. And also the mean cell volume is reduced because these platelets are smaller than red cells, so it's being counted as small red cells. So in this situation, in fact, the hemoglobin was the only reliable reflection of how anemic the patient was. Had we done a manual hematocrit, that also would have been reliable. But just to show you the problems you can have with flow cytometers. This is obviously an exaggerated case, but to a greater or lesser extent, you can see this in animals with larger platelets and smaller red cells. So what about the differential count that you obtain from impedance analyzers? How is this performed? The different analyzers vary in the methodology here. Sometimes they have cell-specific lysing agents, which lyse the different cells and then count the remaining ones. Or they have reagents in there which shrink the cells at different rates and enable the cells to be counted in that way. These have a variable degree of accuracy. So if I would say that if the white cell count is raised or the differential is at all unusual, it would be best to follow up with a blood film examination and a manual differential count. Okay then, so let's move on and discuss how flow cytometers work. And as we've already said, we have a couple of in-practice analyzers, the ProCyte and the LaserCyte, which are produced by IDEX. And then we have some larger commercial laboratory analyzers. So this is a